I'm Dustin Hardwick, a physical therapist with a PhD in movement science. In this episode of the Movement PhD, we're going to take a concise look at muscle, the engine of human movement. We'll review muscle architecture, the surrounding connective tissue, how muscles are innervated, and the basic mechanics of contraction. By the end of the episode, you'll have a clear framework for understanding how muscles are organized and how they produce force. This foundation will support our deeper dives into specific regions and movement patterns in future episodes. On YouTube, you'll see slides appearing in sync with the audio. If you're listening on another platform, the visual aids and full transcript are available on my Substack. Whether you prefer listening, watching, or reading, offering all three formats supports better attention and a deeper understanding of the material. Check the episode description for the links. Now that we have our slides pulled up, let's get started with Season 1, Episode 3, Introduction to Muscle. Let's start with an anatomy overview of skeletal muscle. Muscle is attached to bone via tendon, and that intersection between that muscle and that tendon, that's the musculotendinous junction. Muscle itself is organized into different layers, and there's some connective tissue between those. First, we have the whole muscle itself. The next layer down is a fascicle, a muscle fascicle. That's a bundle of muscle fibers. Those muscle fibers, those represent the cells of muscle. That's where the cell of the skeletal muscle is. So those muscle fibers, they themselves are composed of myofibrils, which are long rows of myofilaments. Those filaments being myosin, your thick filaments, and actin, your thin filaments. So once again, organization from the largest down to the smallest. We have the muscle itself. We have muscle fascicles, the next layer down, which are bundles of muscle fibers. We have muscle fibers, the cell of skeletal muscle. Below that, we have these myofibrils, which are those long rows of myofilaments. Next, let's talk about the connective tissue layers that separate muscle into those different layers. So there are 3 different layers of connective tissue. And those connective tissues not only separate muscle into those different components, but they also have influences on the mechanical properties of muscle itself. I'm also going to talk about these 3 layers like they are distinct entities, but realize they blend in together, so you really can't separate them out within muscle. So they're continuous with one another, and they're also continuous with the tendon at that musculotendinous junction we talked about in the previous slide. So what are the 3 layers? We have the epimysium. So I know this might be new terminology for some of you all. So let's break down the word. Epi just means outside. So epimysium is connective tissue that surrounds the entire muscle. So epimysium is the most outer layer of connective tissue within muscle, which you see is enveloping this whole muscle itself. So one more time, epimysium, epi is outside. So this is the outside layer of connective tissue. Next, we have perimysium. Peri just means middle. So this is connective tissue that surrounds each of those muscle fascicles because it's not on the outside, but it's not all the way on the inside. So it's in the middle, perimysium, surrounding those muscle fascicles. Finally, we have endomysium, which surrounds each of those individual muscle fibers. So it surrounds those cells. Because of that, it is the deepest layer of connective tissue. And guess what? Endo means inside. So this is connective tissue on the inside. So endomysium is the deepest layer, and it surrounds each of those individual muscle fibers. Let's now go through each of those different layers and give some additional details. I've also included some cross-sections of the different layers so you can see what they look like in the tissue itself. Starting with epimysium, epi meaning outside, so this is the connective tissue on the outside. It surrounds that entire muscle. It's composed of dense, irregular connective tissue, which is primarily type 1 collagen, and it has a few important functions. It's important for passive tension, so as you stretch a muscle to its more stretched position, this epimysium provides some tension, and that tension helps to support the joints. It also helps to transmit forces from the muscle itself to that tendon, so it has a really important role in transmission of forces both passively and actively. It also separates muscles within a compartment and reduces the friction between them. So in a lot of parts of our body, there are multiple muscles that occupy a very similar space. We need some way to separate out each of those different muscles and allow them to contract and slide across one another. That's a role of epimysium, is to separate those muscles and to reduce that friction as they both contract. The next connective tissue layer is that perimysium, which just means middle layer. So peri again means middle. So it's perimysium because it surrounds each of those muscle fascicles. There is a typo on this slide. This should say perimysium surrounds groups of muscle fibers forming fascicles. It's made of, again, dense, irregular connective tissue, but now we're talking type 1 and type 3 collagen. And these fibers are arranged in a wavy pattern. That way it has a little bit of give and allows it to stretch a little bit within those fascicles themselves. So what does it do? It transmits forces between those different fascicles. That way you have a smooth muscle contraction. So each of these fascicles can contract together and produce a nice smooth contraction. It's also important for providing structure for the blood vessels and nerves. So that lattice work of wavy fibers, it gives a place for those nerves and those blood vessels to be housed in, to provide some structure for them so they stay where they're supposed to. And our final layer, endomysium. This is, again, the innermost connective tissue layer, and it surrounds each of those muscle fibers. So it surrounds those cells of the skeletal muscle. Again, thinking about the word endo means inside. So this is the deepest layer of that connective tissue. So what does this deepest layer do? Well, it contains a lot of the capillaries that allows for metabolic exchange to occur. It also functions to insulate each individual fiber from one another. So each fiber is electrical activity is isolated. So it keeps that electrical activity during an action potential within each of the fibers themselves. Before we start talking about how muscles contract, let's briefly go over how a signal comes from the brain all the way down to the muscle. So later in this series, we'll have a deeper discussion of motor control and how the brain initiates that signal. But let's just talk high-level overview right now. So the signal starts in the primary motor cortex, and through that upper motor neuron, it's going to send that signal from the brain all the way down to the level of the spinal cord that's appropriate for that muscle. So that whole pathway from primary motor cortex down to the spinal cord, that's the upper motor neuron. So keeping that separate from lower motor neuron is going to be important later when we talk about disorders of upper versus lower motor neurons, because they have different functional implications. So again, let's say that one more time. The upper motor neuron is from your primary motor cortex all the way down to that level of the spinal cord. So once we get down to that level of the spinal cord, there's some interneurons and some things that are going on. But finally, that lower motor neuron exits that spinal cord and sends the signal to the appropriate muscle. So again, your lower motor neuron is from the spinal cord to the muscle itself. So let's review that one more time, just because it's an important concept. Upper motor neuron from brain to spinal cord. Lower motor neuron, or peripheral nerve is another way to say that, is from spinal cord out to the muscle itself. Once that lower motor neuron, or that peripheral nerve, synapses with that motor end plate there at the muscle, that motor neuron depolarizes the muscle fibers, which causes calcium to be released. This calcium, now inside that cell of the muscle, binds with troponin, which then changes the structure of that tropomyosin strands, and it allows that myosin to attach to the actin and pull those actin filaments towards the midline. We'll take a deeper dive into that contraction in a few slides, but this is just an overview. So let's review that one more time. Primary motor cortex down to spinal cord through that upper motor neuron. Lower motor neuron from spinal cord to muscle itself to that motor end plate. That action potential releases calcium, which changes the structure of some proteins within the sarcomere itself and allows the muscle to contract. Let's talk about some structures that are involved in that action potential, that release of calcium that we just talked about. So remember that a muscle fiber is a long cylindric cell with multiple nuclei. Along the outside of that long cylinder, we have our sarcolemma. Another word that you might see is myolemma. They mean the same thing. It is a plasma membrane that fuses with that tendon at that musculotendinous junction. This is where those action potentials are generated. So that lower motor neuron sends that signal down to that motor end plate. That's where these action potentials are generated within this sarcolemma. But this is just on the outside. So how do we get that action potential from the outside of this cell to deep within it? Well, we have a transverse T-tubule, which is basically part of that sarcolemma that dives into the cell itself. So it allows that action potential to get deep within that cell. So how do we release calcium? Well, we have stores of calcium both on the outside and surrounding those T-tubules. The sarcoplasmic reticulum, it stores and releases calcium on that sarcolemma, on that outside. Our terminal cisternae, they surround those T-tubules, and they form this thing called a triad. So let that help you here a little bit. Triad means 3. That's a transverse T-tubule with 2 different terminal cisternae. So that action potential gets deep in the cell from the T-tubule, and it releases the calcium from each of these terminal cisternae. So these are enlarged areas of that sarcoplasmic reticulum. So let's go through that one more time. We have an action potential propagated along the sarcolemma. It gets deep in the cell through these transverse T-tubules, and this action potential releases calcium from the sarcoplasmic reticulum on the outside and the terminal cisternae, which run along those T-tubules to get deep within that cell. And that's what allows that muscle contraction to occur. Keeping with our discussion of innervation of muscle, within a motor unit, and a motor unit is just a lower motor neuron, and all of the muscle fibers that it innervates, they follow the all or nothing law. So what that means is when there's a stimulus, it doesn't matter the strength of that stimulus below or above the threshold, it's either on or it's off. So within a motor unit, it's binary. It's either no response of the muscle at all, or it's full response of the muscle. So any stimulus strength below threshold, that motor unit, those muscle fibers, they're off. They're not contracted. If we cross that threshold, then those motor units fire maximally. All those muscle fibers contract maximally. So again, the all or nothing or all or none law states that within a motor unit, when a stimulus causes a response, all of the fibers within that motor unit fire maximally. It's either on or it's off. That's why it's all or nothing. Below threshold, nothing. Above threshold, it is all. Full strength contraction. Next, let's talk about how we go from a twitch to a nice fused muscle contraction. So a twitch is a single action potential. So it reaches threshold, those muscle fibers fire, and then they relax. If we increase the speed by which we cross that threshold, we'll first get to unfused tetanus. And what that means is we get a twitch and it starts to relax, but then we fire it again before it fully relaxes. And then we do that a few more times so that we're starting to sum those twitches to a higher relative tension, but we're not yet to a smooth muscle contraction yet. When we keep increasing the rate at which we activate those muscles, eventually we'll get to a period where you can't even see the twitch itself. We have a nice, smooth muscle contraction. That's fused tetanus. So let's review that one more time. One single action potential and we let it relax, that's a twitch. We start to increase the rate at which we activate that muscle and we don't let it fully relax before we cross threshold again, that's unfused tetanus. So we start to sum these twitches into a greater relative tension. And finally, if we keep activating it fast enough, you won't even be able to see those individual twitches. That's going to be our fused tetanus or a nice smooth muscle contraction. If motor units are either on or they're off, well then how do I grade the force within a muscle? If everything is either 0 or 1, how do I go from a light contraction when I pick up just my hand or a light object to picking up a heavy object? Well, we grade muscle contraction by motor unit recruitment, by recruiting different types of motor units and different types of muscle fibers that are attached with those motor units. And this is Henneman's size principle. So what that states is, as we increase the force demand of our muscle, we're going to first activate those slow twitch type 1 fibers. So those slow twitch type 1 fibers are the fibers that have a lot of endurance, but they don't have a lot of force generating capabilities. However, they're pretty fatigue resistant. So that is a good thing, because these are always going to be the muscle fibers that are activated first. And that's a good thing because we want that low level activation to be the fibers that are most resistant to fatigue. As we increase the demands of that force, so as we're increasing force, which is on our horizontal axes on a graph that we have here on the slides, next we're going to activate those intermediate fibers. Those intermediate fibers, they have some properties of a slow twitch muscle, so they have some fatigue resistance, but they have more force generating capabilities. So they're an intermediate fiber. So it makes sense that we go from a slow muscle fiber to an intermediate next. As we continue to require greater and greater amounts of force, so we're going from a cup of water to a dumbbell in my hand, now I need maximal force from that muscle. Now I'm going to activate those fastest twitch fibers, those type 2B, 2X fibers. So that's how we grade muscle force. We activate those slow twitch fibers first, and they're going to be activated more often. So that fatigue resistant capabilities of those fibers is going to be a good thing. Then we activate those intermediate fibers, and finally, we activate those fastest twitch muscle fibers, those ones that can generate the most force, but they fatigue a little bit quicker. So this Henneman size principle is how we go from within a motor unit, all or none, to a whole muscle. We can grade that force from a light contraction all the way up to a full maximal tension, 1 rep maximum type of contraction. Now that we know how muscles get their innervation, we know how they grade that muscle contraction, let's go back and talk about some more anatomy. Let's talk about the sarcomere itself, which is our basic functional unit of muscle. And what gives skeletal muscle that striation, so those alternating patterns of light and dark. So that's the one reason why skeletal muscle is called striated muscle, because we have this distinct banding pattern or striation within those sarcomeres. So what are our major components? We have our actin myofilament. These are our thin filaments. It's attached at the Z line. Another way to say that is Z disc, because disc is three-dimensional, but that Z line, that's the demarcation between each of the individual sarcomeres. And during contraction, those actin filaments move towards the middle. So that sliding cross bridge is those actin filaments moving towards the midline. Your myosin myofilament is the thick filaments. These are the motors for the sliding cross bridge theory. They provide the molecular motor that converts that chemical energy into mechanical energy that allows our muscle to contract. These are anchored in the middle at this M line. So let's review these two components because they're the most important parts of the sarcomere. This is why muscles can contract. We have our thin filament actin. It's attached to the Z line, Z disc. And during contraction, it's pulled towards the middle. Your myosin myofilament, that's your thick filament. It's your motor. It's anchored at the M line in the middle here, and it does the pulling. It pulls those actin myofilaments towards midline. Let's briefly go through the cross bridge cycle. So this leaves off where we were talking about the innervation of muscle a few slides earlier. We talked about the calcium being released after the action potential. Well, that calcium, it binds to troponin, which then moves the tropomyosin out of the way on that actin filament. When that tropomyosin moves out of the way, now the myosin head can bind to that actin, which forms that cross bridge. Now that we have that bind between myosin and actin, it releases those inorganic phosphates, which then allows the myosin head to pivot, which is actually the motor part. So releasing that pi from that ADP causes that myosin head to pull that actin filament towards midline. Once that happens, that detaches and a new ATP binds, which causes the head to be released. ATP hydrolyzed again to ADP plus pi, and we recock the myosin head back to its high energy state. And then we start this whole loop again. So it repeats continuously during contraction, which produces that shortening force. So again, calcium, troponin changes tropomyosin, which allows the myosin head to bind to actin. The release of pi causes the myosin head to pull that actin filament towards midline. We detach that. We get a new ATP on there, which is hydrolyzed into ADP plus pi and sets the whole thing back to where it started, and we could start that whole thing all over again. Now that we know how that actin is pulled across that myosin during that muscle contraction, let's talk about what happens to different regions in the sarcomere during that contraction. So let's go through these zones first. So on the outside of the sarcomere, you have that Z disc or that Z line. The area around that Z disc or Z line is the I band. That's the part of the sarcomere that's just actin. That's the light part of the striation of a sarcomere. So I band is actin. The A band is the area of the sarcomere that is the myosin or those thick filaments. That's the dark staining of a muscle sarcomere because it contains that thick filament myosin. So A band is the myosin part. Now there's a subzone within that A band which is called the H zone. The H zone is the part of the A band that's myosin only. So for parts of the A band, there's some actin and there's some myosin. The H zone is the subpart of the A band that is only that myosin. So let's talk about what happens in each of these zones during a concentric muscle contraction first, and then an eccentric muscle contraction next. At the largest level, the Z discs, they get closer because the actin is pulling those Z discs towards the middle. So the distance between the Z discs or C lines will get smaller during a concentric muscle contraction. Similarly, the I band, which is the zone of actin within a sarcomere, is also going to get smaller because, again, those actin are being pulled across that myosin, so that I band is getting smaller too. The A band, the zone for the thick or myosin myofilaments, it doesn't move because it's the motor. It's the one that's anchored in place and pulling that actin towards it. So that A band's relative length is going to stay the same because it's doing the pulling. However, that H zone, the area that's just myosin, that's going to get smaller because it's the zone from one actin filament to the next. So if you pull those actin filaments towards the midline, that H zone is going to get smaller. So that's represented by this graph up here. Z lines get smaller, A band stays the same, H zone gets smaller, I band gets smaller. The opposite occurs during an eccentric contraction. Because an eccentric contraction, it's a lengthening contraction. Therefore, those Z lines are getting farther apart. The I band also getting further apart. That H zone, similar to those two, getting further apart because that actin is being pulled farther away. Again, A band stays the same. So your A band is your one constant because it's the motor, and all the rest of them will follow the contraction type. Here is an image showing those different zones moving during a concentric muscle contraction. You can see the Z disc getting closer, the H zone getting smaller, the I band also getting smaller, whereas that A band just stays the same throughout. Let us now briefly discuss the blood supply to skeletal muscle because we need to get energy into the cell. So that's what the role of blood supply is to bring energy to the cell and carry those waste products away. At the highest level, we have arteries, which carry oxygen-rich blood to the periphery. Next, we go down to the arterioles, which are the smaller layer of the arteries. Those are going to deliver that oxygen to the muscles themselves, which these are at the level of the fascicle, these arterioles, and that lattice work within that perimysium that surrounds the fascicles. That's where you'll find these arterioles. And they're going to deliver the blood to your capillaries, which are around those muscle fibers themselves. The capillaries, they're thin-walled, and they allow nutrients and gases to pass in both directions. So oxygen to the cell, and then waste products back from the cell to the capillary itself. So from there, those waste products will go to the venules, which are in that same level as the arterioles. So they're within that perimysium that surrounds those muscle fascicles, carrying that oxygen-depleted blood away from the muscle itself. And finally, to your veins, which carry your oxygen-depleted blood to the lungs themselves to reoxygenate. The skeletal muscle pump is one way in which we can assist that venous return or getting that waste product from the muscle back to the heart and lungs. This is really important in gravity-dependent body parts. So think about your calves and your legs. They're working against gravity. So this muscle pump is what's going to allow that blood to get back to the heart, and it can generate about half of the total energy needed in getting that circulating blood back to the heart during exercise. So how does this skeletal muscle pump work? There are one-way valves within these veins, which keeps the blood from going backwards because we wouldn't want the muscle to push it back down, so it can only pass towards the heart. So as you walk, you go on your toes, you do like a knee pump or any kind of thing where we're contracting these calf muscles, they're going to squeeze down those veins of the lower limb, and it's going to push that blood back towards the heart. And that's the reason why if you're standing in hot weather, not moving, people tell you to go up on your toes, to unlock your knees. All of these things are going to activate these muscles, which then allow that deoxygenated blood to pass back towards the heart. Previously, I mentioned the Henneman size principle, by which we activate the slow twitch fibers first, all the way up to the fastest twitch fibers next. Let's kind of go through that real quickly. This is a high-level overview of the different skeletal muscle fiber types. And realize this is oversimplified. There is an infinite number of morphology from slow to fast twitch, but we're going to break it into 3 just for simplicity. So our type 1, or our slow twitch fibers, their contraction velocity is quite slow. So it takes them a long time to get to full tension, 100 milliseconds for some of them. Force generation is going to be low. These are smaller motor units. They generate low tension, but they're really fatigue resistant. So think about activities that activate your type 1 fibers would be your endurance activities, walking for hours, standing. Your postural muscles that are active to keep you upright, they're going to have a lot of type 1 fibers because they're highly fatigue resistant. Metabolic properties, they're highly aerobic type. They have a lot of mitochondria because of that, and they have a rich blood supply because they're taking that oxygen in from the blood supply. We'll talk a little bit more in future units on your nerve axons and motor units, but realize these are our smaller diameter motor units that activate these type 1 fibers. So again, our primary function, postural maintenance and long duration, low load types of endurance activities. Let's jump to the fastest twitch next, and then we'll come back to our intermediate. Our type 2X or our fast glycolytic fibers, these are very fast to peak tension, about 4 times faster to peak tension than your type 1 fibers, approximately 25 milliseconds. They have large force generating capabilities. These have huge motor units, and they can develop lots of tension, but they run out of energy quickly. So they have low fatigue resistance here. So they're going to fatigue quite quickly. These are anaerobic muscles, so they don't need a lot of mitochondria. The nerve fibers that activate these fastest twitch muscle fibers, these are our largest motor units. They're going to have the largest diameters. And because of that, they're going to be activated last. So remember that Henneman size principle, from smallest to largest, these are going to be our largest motor neurons. So they're activated last. These are for powerful movements. Think about jumping, sprinting, 1-rep max types of things for these type 2X. Then we have these type 2A. I like to just call them intermediate fibers because there's a whole bunch of intermediate fibers between our slowest and fastest twitch fibers. Some of them are closer to slow twitch. Some of them are closer to that fast twitch. They have an intermediate force generating capabilities. So they're in the middle for contraction velocity, in the middle for force generation. They have a moderate amount of fatigue resistance, and their metabolic properties are somewhat between the two. So some properties are aerobic, some are a little bit more anaerobic. They could really represent the gamut between the two. And our nerve and motor units, they're going to be medium because they're activated between those slow twitch and those fast twitch fibers. So Henneman size principles, these are going to be the second level of muscle fibers that are going to be innervated. So these are going to be moderate activities. Walking, running at kind of higher pace than just casually doing it, which would be a lot of slow twitch fibers. Let's talk about how fibers are arranged in muscles themselves. And there are 3 major types of muscle fiber architecture. Our fusiform muscle, another way to say that is a parallel muscle, These have long fibers that go from origin all the way to insertion. So they run the length of that muscle. A fusiform muscle or a parallel muscle, they are optimized for range of motion because when they contract, they pull that insertion towards that origin across a longer range. So examples of our fusiform muscles, bicep is the easiest. You can easily see your bicep. Those fibers go all the way from origin to insertion. Your bicep is designed for excursion or range of motion at the expense of force generating capabilities. So fusiform muscles have the least force generating capabilities, but they can do it across a longer range of motion. A pennate muscle, now we have multiple fibers that insert on a central tendon at an angle. And because of that, we can pack more muscle fibers into a muscle of the same size. So rec fem or rectus femoris of your quadriceps is a great example of a pennate muscle because you have all these fibers that converge on a central tendon, and it allows that rectus femoris to have a lot of fibers in it, more fibers than if it was a parallel. So it can generate a lot more force. But that force is not going to be over as far of an excursion. So pennate muscles designed for force generation, and they give up some excursion for that. And the thigh is actually a good example of two different muscles with different fiber types that run close to one another. Your rec fem is a great example of a pennate muscle, where your sartorius is a great example of a fusiform muscle. And you think about what each of these does, your sartorius has to do abduction, external rotation, and flexion at both the hip and the knee. So it needs that fusiform muscle to be able to contract across all of those different joints. So that excursion is needed. Whereas your rec fem, you need the power for that knee extension from your rec fem. A convergent muscle, and another way to say convergent is fan shape, is a muscle that has a broad origin, but then goes to a relatively small area of insertion. So a great example here is your pec. You have origins across the clavicle and sternum and some slips to your abs, and then they all converge on the humerus. So that converging muscle pattern, the pec is a great example. Another example would be your glute med, glute min of your hip. So your hip abductors are also great examples of convergent muscles. Here's a table that summarizes the difference between these three. So again, fusiform or parallel. The fascicles run parallel to the muscle's long axis. They are really long fibers, really good for range of motion and speed, but their force generation is a little bit lower because their cross-sectional area is not as great. Example again, biceps. Pennate, feather-like. So another way to think about pennate is a muscle that looks like a feather. These have short fascicles and they attach at an angle to a central tendon. There are some subtypes, and we'll talk about these in the individual units. Unipennate just means they attach on one side. Bipennate just means that they look like a feather, so it attaches from both sides. Or multipennate means that there's a bunch of different pennate angles. Deltoid would be an example of a multipennate because you have three different heads that all have some pennation to each of those central tendons. So high cross-sectional area, high force generation, but the contraction speed and range is lower because they're at an angle. So rec fem is a bipennate, deltoid, multipennate. Convergent, broad to narrow, fan shape, these are all saying the same thing. Broad origin, converging on a single tendon. Versatile pull direction. So because of that, we can use a convergent muscle to move across several different directions. So think about your pec, which has roles in horizontal adduction, flexion, and extension, depending on where your humerus is. It needs that convergent muscle to be able to do all of those things. Similar to glute med, besides just doing abduction of the hip, it can do flexion, it can do extension, it can do internal and external rotation. And that's because of that convergent or fan shape orientation of those fibers. So again, pec major and the glute med, glute min being another example of that. I mentioned cross-sectional area. There's two different types of cross-sectional area, and both of them have different rules for measuring different types of muscles. So our anatomical cross-sectional area, that's just taking a muscle and cutting it across its widest part and counting the fibers there. In a parallel muscle like your biceps, you're going to get all of those. However, if you cut at the widest point of a pennate muscle, you're not going to get all those fibers because they're short fibers that attach at an angle, so you're going to miss a bunch of those fibers if you use an anatomical cross-sectional area. Therefore, an anatomical cross-sectional area is able to get the cross-sectional area of a parallel muscle, but it underestimates pennate muscles. So it underestimates the size of a pennate muscle. It underestimates the ability of a pennate muscle to generate force. So what do we do about that? We have a different kind of cross-sectional area that we use for pennate muscles. The physiological cross-sectional area is the area where all the fibers are present. In a parallel muscle, physiological cross-sectional area and anatomical cross-sectional area, those are equivalent. They're the same thing. That cut across the middle will get you all the fibers. However, in a pennate muscle, now we have to cut through all of those fibers. Here it is in a unipennate. Here it is in a bipennate type of muscle. You can see that that cross-sectional area is much larger than the anatomical cross-sectional area and really gives you an idea of how many fibers are present in that muscle. So this physiological cross-sectional area is how you measure the cross-sectional area of a pennate muscle. Anatomical cross-sectional area works for parallel muscles. Next, let's talk about the length-tension relationship of a sarcomere. This is an important concept, and we're going to come back to this in each of our units and talk about how putting muscles in different positions impacts their ability to activate and produce force. The reason, this length-tension relationship of the sarcomeres. So at resting length, so at the mid part of the length of a sarcomere or a muscle, this is where we get the optimal amount of tension generated. And that's because at that resting length, all of the actin and myosin myofilaments are in contact, so I have good cross-bridging, but I still have room for that actin to slide. So I have good cross-bridging and I still have room to slide. So that makes that midpoint of that length of the sarcomere optimal for generating tension. However, if I'm on that ascending part of the curve, where that sarcomere is shortened, I have good cross-bridging, but I have nowhere to go because I have those actin filaments are already maximally shortened, so there's full overlap. There's nowhere for those filaments to go. So when it's fully shortened, I have nowhere to go. So I'm generating no force when a sarcomere is over-shortened. Conversely, when we are maximally lengthened, so we have a muscle at the end range, so all of the sarcomeres are as lengthened as they can, I have very little actin overlap with that myosin. So not very many of those motors can do their job. So let's review that. Maximally shortened, I have good cross-bridging, but I have nowhere to go. Resting length, I have good cross-bridging, and I still have room to slide, so maximum tension is generated near that resting length. Maximum lengthening, I don't have very good cross-bridging because not very much of that actin is overlapping those myosin. So this is an important concept. Review this several times. I will review this concept as we go through the different units as well. The active component of a sarcomere is not the only thing that affects the overall tension of a muscle. That intramuscular connective tissue, and we all remember what that is, right? That's that epimysium outside, perimysium in the middle, endomysium at the lowest level, also titin, which is a connective tissue within the sarcomere itself. These connective tissues also contribute to the properties of the muscle. So as we first elongate a muscle from kind of shorten to lengthen, the very first part of that tension on that connective tissue, it's just going to align the fibers. So at rest, those connective tissues have a little bit of a wave in it. That initial tension on that connective tissue just aligns those fibers. No tension yet. As we continue to lengthen the muscle and put tension on those connective tissue, that tension generates the first part in the elastic region, then through our plastic region. Hopefully we don't get to the very end range. So we'll talk about the stress strain curve in a bonus episode in the coming weeks. So what's the function of that connective tissue, that passive tissue properties of that connective tissue? At that extreme lengthening of a muscle, we don't have very good active component. That passive tissue tension on that connective tissue can support the muscle, can support the joint. So at the very end ranges, again, that sarcomere is overly lengthened. We need to have something to support that joint, to support that muscle, that connective tissue from that peri, epi, and endomysium is there to do that. Let us talk briefly about strength change across the lifespan. Muscle mass and strength, they reach their peak in the 20s, a little bit younger for female, a little bit older for male. And it's followed by a gradual decline from midlife on. Again, these are studies looking at untrained individuals. So obviously your muscle mass and strength might not peak in the 20s if you don't start training until your 30s. But for an untrained individual, yes, we see a peak of that strength around 20 years of age, 20 to 25 years of age. The rate of decline is minimal up until 50 to 65 years of age, which we see a steepening of that rate of which the strength is declining. So this strength loss related to age is observable in both cross-sectional, just taking groups of people, which is one level of evidence. Also in longitudinal studies where they follow older individuals from one age to an older age, we see that age-related across both. So that's giving us good evidence that there is an age-related loss in the muscle strength, the force-generating capabilities of that muscle. All strength domains are affected. So we see losses in power, in strength and function and endurance. However, the strength loss is greatest for those 1 rep max power types of activities because as we age, we have selective denervation of those largest motor units. So selective denervation of those 2X fibers and the motor units attached to it. There's a preferential sparing of those type 1 and some of those intermediate fibers that are more type 1. Therefore, you can maintain your endurance for much longer in life than your strength and your power. And if you look at sports, you can see that play out. Activities that are a lot of power, a lot of strength, those are activities where younger individuals do best. However, if you look at the entrance for a ultra endurance type of event, you see a lot of individuals in their 50s, in their 60s that are able to compete with people much younger than them. And that's because they're sparing those type 1 fibers. Loss in all domains can be mitigated by regular exercise. So even that power component, that strength component, with regular exercise, you can make that loss much less. You can't get rid of it completely because that denervation is a real thing, but you can mitigate a lot of that strength loss with regular exercise. And you can maintain most of that endurance. That also requires keeping your cardiopulmonary function as well. So realize that endurance type activities, it's a little bit of muscular endurance, a little bit of cardiopulmonary health. So maintaining the health of that system would be important as well. Sex differences in strength. Males typically have a higher absolute strength across all age groups, but it's just a size thing. When you normalize muscles to the body mass, the differences go away, especially in the lower extremity. This graph shows you glute max cross-sectional area just as a whole muscle. When you normalize it for the cross-sectional area of the glute max, that changes to where this is not a significantly different graph here. So what it means here is that muscle is muscle. It only differs in the amount of muscle. It doesn't differ in the properties of the muscle itself. Some evidence suggests females may experience a greater relative strength loss across ages, and there are some hormonal reasons for that, but they also respond best to resistance training later in life, so they can make up that gap with regular exercise. So practical implications here, when you're looking at strength between the sexes, you're only going to see that difference on absolute terms when you're trying to move a 1 rep max of an external load. For body weight activities or activities that are normalized for body weight, you will not see a difference between males and females because muscle is muscle. It doesn't matter who's wearing it. Now let's talk about the effects of a prolonged stretch or prolonged shortening on the muscle itself. So what do I mean by prolonged lengthening or prolonged shortening? So a lot of this could be posture. I'm going to use that as my example for today, and we'll talk about some of the other examples later on. So many of you are watching on a computer, watching on an iPad, listening on a phone. Chances are you might be seated. You may be looking down at your device. If your posture is not very good, The muscles on the backside of your body, so think about those muscles that produce extension of your spine, they might be lengthened because you're hunched over into your device. So those muscles are at a prolonged lengthening because you're doing it for hours. Think about muscles on the other side, they're just going to be shortened. So the muscles that cross the anterior part of the hip, so your hip flexors, if you're seated, if you're driving, those are going to be under a prolonged shortening. So what happens in each of these muscles as you're listening to this podcast or studying something else? For those muscles on the posterior aspect that are under a prolonged lengthening position, they're adding sarcomeres in series. So they're increasing the number of sarcomeres within each of those fibers and making those muscle fibers longer. So what does that do? It changes where that muscle is able to generate its best force or most appropriate force. So prior to your studying over many weeks or months, your muscles that keep you upright, they might be in their best position when we're sitting upright, just like this. So they're right at the peak of their length tension relationship, and they're able to produce maximal force. I've been hunched over studying for months on end. I've added those sarcomeres in series. Now the position by which those muscles are able to produce most force is in some of that hunched over. So now when I try to straighten up, that muscle is no longer at its peak and it's not able to generate as much force. So prolonged lengthening, it leads to hypertrophy in series, but that changes that length tension relationship of all of those sarcomeres, which means that muscle might not be able to function optimally in its normal ranges. The opposite happens in those muscles that are under a prolonged shortening. What does it lead to? Chronic shortening lead to atrophy and loss of sarcomeres in series. So now I have less sarcomeres in series, so it's making that muscle shorter. So now when I try to get through my normal ranges of motion, I either cannot get there. So now I have tight hip flexors. I'm unable to get full hip extension, and I'm getting extra tension within those normal ranges, which can lead to pain and significant impacts on function. So prolonged lengthening is bad. Prolonged shortening is also bad. We'll give a lot of examples throughout this podcast when we start to talk about postural effects on different muscles. So understand the concept right now. We'll apply it in some future episodes. Let us talk about the implications of relative activity on muscle performance. So if you look at this graph here on your vertical axes, this is the relative amount of activity you're doing. So relative physical stress. So how much activity are you doing in your daily life? Hopefully you're somewhere here in the middle. If you're in this kind of lower middle part, that's maintenance. You're not doing more than you usually do. You're not doing less than you usually do. So you're maintaining your muscles about where they are. So you're not losing muscle, you're not gaining muscle, you're just in a period of maintenance. Or you might be in this hypertrophy range. You are regularly exercising, so you are gaining some muscle, but you're doing it at a rate that's healthy, so your body's able to adapt to that exercise. So that's our hypertrophy zone. These two zones are where the body's most healthy, either maintaining what you have or adding some muscle. If you get outside of these zones, that's when things become less healthy. So if you are now studying all the time and you're not working out, you're not going for a walk, you're just listening to my podcast and studying, and you're much less activity, you're going to get some atrophy. So your muscles will start to break down because your body doesn't like to have a bunch of active tissue that it has to maintain. So it starts to lose some muscle. The other end of the extreme, you've been in this maintenance zone and you decide that I'm going to run a marathon in 2 months, and you don't run. So you're starting from zero and you start a marathon program. Now you are doing too much activity and your body can't adapt to it. So you're at high risk for injury, stress fractures, overuse injury to muscles, other types of injury to different tissue. And if you really overdo it and you can't keep up with your nutrition, that can also lead to death, either of the tissue or the whole body. Thank you for joining me for this introduction to muscle. We reviewed muscle architecture, connective tissue layers, innervation, and the essentials of contraction. These concepts will be important as we move forward. If you found this helpful, please subscribe to the podcast and follow me on social media. And if you have questions or specific topics you would like to see covered, Let me know in the comments. In our next episode, we will begin our hip unit with an overview of hip anatomy and movements, a perfect transition to applying today's muscle concepts to one of the most powerful regions of the body. Thanks again for listening, and I'll see you in the next episode.